Hey gang, Matt Greer here. Today I will be giving my presentation on Mendelian Genetics. The objectives for this are I'm going to cover the basics of genetics, we're going to talk about Punnett Squares, Pedigrees, and some human disease. Now hopefully by the end of this PowerPoint, this presentation, my students will be able to understand how these work and to use them to solve simple problems. My name is Matthew J. Greer. This is prepared for Biology 510, a comprehensive overview of cell and molecular biology through Grand Canyon University. And my professor is Dr. John Wooll. This would be taught to AP Biology or AP Anatomy and Physiology students, and is an important part of their curriculum to understand genetics and to talk about those kind of topics. So without further ado, let's get started. So what is genetics? Genetics is the branch of biology that studies genes, genetic variation, and heredity in organisms. For example, this giraffe here, courtesy of Brady, is an albino giraffe. This is a genetic variation that caused this giraffe to look very different than a normal giraffe does. Prior to the 19th century, the common thought on genetics was that an organism acquired its traits through this blending of inheritance. And this is where mom and dad contributed to your genetics, and the average of those contributions was that you ended up looking like. This theory was the predominant theory until the work of Gregory Mendel. Genetics now, we can use it to describe several human diseases, and we can talk about how they're passed from parents to offspring and the odds and the probability that those genes will be passed on. It's a very exciting field of biology. So who was Gregory Mendel? Gregory Mendel was born in 1822 in what is now the Czech Republic. Early on, Mendel had a lot of success in physics and mathematics. His parents were very poor and couldn't provide money for an education. Luckily, his professor suggested him to the Augustinian monks. These monks were held education very highly, and because of his work at the abbey, he was allowed to keep studying at Vienna and learn statistics, which would come in very handy. His success came from being a meticulous note taker. Mendel kept really good notes. He kept a lot of notes. This is very important when you want to look back through your data. Good science is good note taking. We call Gregory Mendel the father of genetics, but he didn't set out to crack the code on genetics. He set out to create a hybrid pea plant. In the process, he noticed some trends with these plants, and he noticed that some things kept happening over and over in a very predictable way. Mendel very quickly noticed that not all pea plants had the same traits. In this image here on the right, there's some round pods on the left and some wrinkled pods on the right. Very different looking peas. Mendel also found that some of his plants were true breeding, and he bred many of them to be true breeding. And these are pea plants that when bred with a plant of the same type, example, a round pod bred with a round pod, these always produced offspring with that trait. In this case, the round pod. Mendel's methods. Mendel carefully controlled pollination between pea plants. He would cut the other flowers off so that he could control himself the pollination between pea plants. Altogether, he looked at 7 different unique traits for peas. He looked at seed shape, seed color. He looked at the pod shape, the pod color. He looked at the flower color. He looked at the flower location. Was it axial, like off to the side, or was it terminal on the top? And he looked at plant size, whether they were tall plants or short plants. With all these crosses that he did, he kept meticulous notes on these. And from this data, he could make some conclusions. The results of Mendel's experiments. Mendel noticed that some factors were hidden by others. He called the factors that weren't hidden, these factors that came to the forefront all the time, he called them dominant factors. And this is a trait that only needs one copy to cover up the recessive trait. So in this picture on the right here, let me grab my pointer, the purple flowers, when bred with white flowers, always produced these purple flowers. This purple trait was said to be dominant because it's covering up that white trait. Now recessive is a trait that needs 2 copies to be observed, and it hides normally under the dominant one. So these 1/4 white on the bottom, that's a recessive trait because it got lost in this first cross. Now we're going to talk a lot about genetics. So we're going to talk about phenotype and genotype. Genotype is the genetic combination of the factors, like what the actual DNA says, and phenotype is the physical appearance of the organism. And these can be similar but different. The law of segregation. At fertilization, each parent's gamete gives one copy of a trait. In this manner, all sexually reproducing organisms get one copy from mom and one copy from dad for 2 copies. In humans, we have 46 chromosomes. We describe them as 23 pairs of chromosomes. This is because we get one from mom and one from dad. The law of segregation simply states that 2 alleles for each trait separate during gamete formation and then unite at random during fertilization. Because of this, and because of the statistics behind it, Mendel saw this 3-to-1 ratio, where at the end, when you're breeding these plants together, you get 3 of these purple, these dominant R's, so they only need 1 copy of that, and then these recessive whites, so that 3-to-1 ratio. And this is very important for these crosses. So peas and humans. While we look very different, peas and humans, all sexually reproducing organisms follow this law of segregation. So after studying peas, Mendel tested several different organisms, such as bees and hawkweed. And in fact, some human genetic diseases behave the same way as pea genetics. So Huntington's disease, cystic fibrosis, and sickle cell anemia all behave in this manner. And we're going to talk about those in a little bit. So to graphically organize this data, Punnett created these squares. And we use these to help represent genetic flow from mother and father into offspring. So Reginald Crundle Punnett, he's the author of the book Mendelism, and he created the Punnett Square. Wooll's given credit for it. He also published some work, Mimicry in Butterflies and Hereditary in Poultry. He was very interested in genetics and studied it throughout his life. We use the Punnett Square because it's a very easy method to track monohybrid crosses. These one trait, one factor crosses. Here on the right, I've got an example of one of Punnett Squares. It's a little bit more simplified now, which is okay. I like some of the historical, the lines and dashes, which we don't use as much. I like those. Those are kind of cool. So how to use a Punnett Square? To use the Punnett Square, you need the genotype of both parents. For our first example, we're going to look at sickle cell anemia. This is an autosomal recessive disease. That means that it would hide underneath a dominant normal trait. Now, autosomal, we have 2 types of cells in our body. We have autosomal, which is our body cells, and we have our sex cells, which are our gametes, sperm and egg. So the dominant allele receives an uppercase letter. In this case, we're going to use an uppercase S for sickle cell. The recessive allele receives a lowercase letter. Now, in our first example here, a mother with sickle cell anemia is going to have offspring with a father who carries the gene but doesn't have the disease. And we're going to look at what is the probability that the offspring will have this disease. So mom's genes, we're going to put on top here. She has sickle cell, so she's going to have 2 little s's. And dad's genetics, let me grab a different color here, are going to be big S, little s. Now, how do you use a Punnett Square is you take these alleles and you move them right across in the cell they're in. So for this big S, it goes here, and dad's little s is going to go here and here. Now for mom's genetics, you're going to bring, excuse me, the little s down. All right, so the question is, what is the probability that these offspring will have the disease? Now, to have the disease, you have to have 2 copies of the recessive trait. So this one has 2 copies of the recessive, and this one has 2 copies of the recessive. So overall, we have 2 out of 4, which equals 0.5, or 50% of these offspring have sickle cell anemia. Now, genotype descriptions. When describing genotypes, we very commonly use the terms homozygous and heterozygous. Homozygous is both alleles are the same for that particular gene. So big S would be a homozygous dominant genotype. And in the example of sickle cell anemia, in order to have the disease, you need to have a homozygous recessive genotype, or little s. Now, heterozygous is a little bit different. These genotypes express different alleles for each copy of the gene. So big S, little s would be heterozygous genotype. So let's do another practice problem for Punnett squares. An individual who has sickle cell anemia has offspring with an individual who is homozygous dominant for the trait. How many of their offspring have sickle cell anemia? So we know that sickle cell anemia, the one who has it has little s, and we now know with this new information that the other individual is going to have big S, big S. And let me use a different color for that because that doesn't quite show up. Big S, big S. I'm going to make them really big to accentuate the difference. So just like we did before, we'll take those big S's across. And we'll bring those little S's down. So our question is, how many of the offspring have sickle cell anemia? Well, all of them have the genotype big S, little s, which means that none of them have sickle cell anemia, because sickle cell anemia needs recessive on both. So 0% or 0 individuals. So carriers and disease. We've talked about these people carrying the disease but not having it. These individuals that are in a population that have a copy of the disease yet do not present with the disease, they don't have it. These are called carriers. And with sickle cell anemia, which is quite interesting, the mutation that causes the disease is beneficial only if you're a carrier. If you're a carrier, you are immune to malaria, and this mutation arose and is very common in areas where malaria numbers are high. However, having 2 copies is non-beneficial, and you get these sickle cell blood cells up on this picture above me. Those are not as good at carrying oxygen, and that's a big detriment to a large organism like a human who has a very high oxygen requirement. So dominant disease. We've talked about sickle cell anemia, which is a recessive disease. Now we're going to switch gears and we're going to talk about a dominant disease. So Huntington's disease is an autosomal dominant disease. That means it is a body cell, carried in a body cell, and it's dominant. So having 1 copy means that you'll show the trait. Huntington's is a neurodegenerative disease, so it attacks the nerves and degrades them. And it usually shows up later in life. So individuals who have this disease usually have time to make it to adulthood, create offspring, and pass on this trait. If you have 1 copy of the gene, you're likely to be affected by this disease. So for our Punnett square problem for this one, we're going to blend together this dominant thing and we're going to blend together carriers. So if you have 2 individuals who are carriers and they have offspring, what are the odds that these children will be affected by this disease? All right, we're going to use an H for Huntington's, where we use an S for sickle cell. And this first one's a carrier, so it's going to be big H, little h. And our second one is also a carrier, so it'll be big H, little h. And then like before, we're going to carry those across on this side and down from the top. Now something to note here, little h, big H, and big H, little h are the same thing. They are the same genotype. Just how Punnett squares work, sometimes they end up looking different, but they are the same thing. So what are the odds that the children will be affected by the disease? So we have, it's an autosomal dominant, so it's going to be, if you have 1 copy, then you have it. So this one, and this one. That circle was a little wonky. So altogether, we have 3 out of 4, or 0.75, or if you're a probability person, it'd be 75% will be affected by this Huntington's if 2 carriers produce offspring. Now, pedigrees. While Punnett squares work for individual crosses, they're not so good at looking at like large familial lines. There's a lot of genetic information. You're looking at a lot of different Punnett squares. So we use pedigrees to track traits back over several generations. Now, the generations are going to be labeled using these Roman numerals. So this first one is generation 1, generation 2, generation 3, so on and so forth. If you had 4, 5, 6, however many generations you're going, you just increase 1 Roman numeral each time you move down. So how do I use a pedigree? Pedigrees can show a lot of information. However, some information is common and is usually seen on most pedigrees, and then a lot is you're probably not going to use it on every pedigree. You might be very circumstantial. So we're going to talk about the important ones. So a horizontal line between shapes is going to represent mating, and a vertical line will represent the offspring. Now, what do the actual shapes mean? If it's a square, it means it's going to be a male. If it's a circle, it'll be a female. So in between a mating pair, you should see one circle and one square. Now, if the shape is shaded in, if it's black, then it is an affected individual. If the shape is half shaded, it'll be heterozygous for an autosomal recessive. You also see some really cool nomenclature with twins. You see these cool triangles. They're not really common. We don't see a lot of twins unless you have twins runs in your family. If it has a slash through it, it means that that individual is no longer living. They died. And inside of each generation, while the generations are labeled in Roman numerals, the individuals will be labeled in order of birth. So 1, 2, 3, etc. So with pedigrees, and we can talk about X-linked genes, especially in humans. In humans, the Y chromosome is much smaller than the X chromosome. And you can see that in this image above me. Because of this, a gene on the X chromosome might not have a homologous copy on the Y chromosome. There might be this empty space where this other gene is supposed to be. Because of this, men that have an X chromosome that carries a disease and is in this zone where there's no homologous area will present the disease regardless if it's a dominant trait or recessive. They only have one copy to display. There's no hiding behind a dominant one if it's recessive. So some examples of ailments like this are Duchenne muscular dystrophy, color blindness, and hemophilia. So let's do a sample pedigree problem. We're going to look at Duchenne muscular dystrophy, or DMD, and we're going to be looking at this pedigree. Keep in mind that DMD is an X-linked recessive trait. So if you're a male and you have one copy of DMD, you're going to have the disease because it rides on the X chromosome, and there's no homolog on the Y chromosome. So let's look at individual Roman numeral 1, number 1. This is a male because he's a square, and he does not have the disease because he's not shaded in. So what does this tell us about his genetics? Well, we know he's male, so we know he has a Y chromosome. And for X-linked, you're always going to put the X, and then you're going to sub-notate above it in a subscript whether he has it or doesn't have it. So we're going to use, for Duchenne muscular dystrophy, we're going to use big D as normal and little d as infected. So he is a normal little d. Normal big D, excuse me. So he does not have the disease. Now individual in Roman numeral 2, and individual 2, this is a lady, as she is a circle. She is the offspring of 1 and 2, as you can see by this vertical line. She is siblings to 2, 3, and 4. Now, she does not have the disease either. She has 2 X chromosomes. But we know that her mom, Roman numeral 1, 2 here, had to have a copy because she is this big, solid, afflicted individual. So she had to be at least one little d in there. Now, dad can only give 1 X chromosome. He can only give this one here. And so that one was a dominant normal one. So she does not show the disease. She is, however, a carrier for it. Now let us look at individuals 3, Roman numeral 3, 6. Now, she's going to get her genetics. It's a girl, she's a circle, from her parents. Her siblings also have this disease, but her parents also do as well. So dad only had 1 X chromosome to give. That was a D. And mom could have given either one of hers, and it would have been a D as well. So she has 2 copies of the DMD allele. She has one from mom and one from dad, each one of them riding on the X chromosome. If you were to have an individual who had 1 known allele and the other is unknown, you could denote it as D question mark or XD question mark. Let's look at another sample pedigree problem here. We're looking at albinism. This is an autosomal recessive trait where afflicted individuals do not produce melanin, so they have light skin, they can have reddish eyes, or they can have very light blue eyes. The following pedigree shows the movement of albinism through a sample family. So we're going to do very much the same process we did in the last one. We're going to start looking at Roman numeral 1, number 1. This is dad here. He is not an albino. He does not have albinism. But looking down at his kids, there is one that does. So what this tells us, and let's use a big A and a little a. So he has to have a big A because he does not have this recessive disease. He has the dominant factor. And then he has a little a because he had to pass it down to his offspring. You have to get your genes from your parents. Unless there's some kind of funny business, mailman, milkman, you have to get your genetics from your parents. Now, Roman numeral 2, individual number 3, he has the disease. So his is going to be little a, little a. It's a recessive disease, so he has to have 2 copies of this recessive allele. And our final individual is going to be Roman numeral 4, number 1. This male also has the disease. He's shaded in here. So we know that both of his parents had to be carriers. They both had to be that big A, little a. And you can go through it and find all the genetics from a pedigree such as this if you work forward and backwards. Start with what you know. Start with the people who are affected. Those are very easy to pick out. And then from there, if it's a recessive, they're going to have 2 copies. If it's a dominant, they could have 1 copy, and you're going to have to dig a little bit further into their pedigree to find out which is the case. This concludes my presentation on Mendelian genetics. These are the references I used throughout this PowerPoint. I hope that you feel much more confident in genetics and that you feel that you can properly use Punnett squares and pedigrees to track heredity and genes. These references are awesome resources if you want to go out and learn more about the material. I particularly really like the one by Child, Mendelian Genetics, Patterns of Inheritance in Single Gene Disorders. That has a lot of good information if you're really excited about learning about different diseases and their genetics. Thank you again for watching, and have an excellent rest of your day.